Okay, go ahead and get started. So as Jim mentioned, I'm not Fred. So let everybody know the baby is healthy. Connie, Fred's wife, is healthy. Hopefully they'll be released here soon. Fred can be back in the lab. But hopefully I'll be able to do him and the lab justice. So I'm here to talk to you today about some C. elegans neurobiology techniques using C. elegans. And really what the Hernley lab is focused on is we're looking at glutamatergic transmission, but mainly in the form of glutamatergic transport and how it relates to synaptic plasticity and memory. So just to kind of give a brief background on the history of C. elegans, people that might not know, but C. elegans was first really introduced and utilized in 1963 by Sidney Brenner. And what a lot of people actually don't know is Sidney Brenner worked very closely with Watson Crick, but Rick Brenner actually wasn't the first person to use nematodes or use C. elegans to study research. But there was actually a group in the late 1800s that were actually using Ascaris worms to study different properties about horse gut, actually. But it was really Sidney Brenner that kind of really pioneered the use of C. elegans. And then just 11 years later, Brenner actually published the first mutant study found with C. elegans using 4 genetic screens, which Noreen touched on a little bit, and I'll kind of explain how we do that in C. elegans in a little bit. And then not too long after that, what you guys might know C. elegans are famous for is actually with apoptosis. Howard Horvitz came along, and then not too long after that, we actually had the complete connectivity of the entire nervous system of the worm. And so the worm is very simple. It only has 302 neurons. And again, I'll kind of touch on that a little bit. And so then another kind of famous person you might know is Martin Chalky. So in '94, he was actually the first one to use GFP, and C. elegans was the first animal to be used in. So here in GFP, we basically just have 2 different types of mechanicization neurons that we have labeled. But then Craig Mello and Andrew Fire got a little bit jealous of Martin Chalky's Nobel Prize, so they wanted one as well. So they actually were utilizing RNAi. And using RNAi in C. elegans is extremely easy. All you have to do is just feed them double-stranded RNA that works as a gene silencing. And then again, just a year later, we have the complete genome of C. elegans in sequence. And C. elegans was in fact the first animal to have its entire genome completely sequenced. But how do we use this guy in terms of neurobiology? So C. elegans is an extremely powerful genetic model. It has a very short lifespan. From being hatched to a full-fledged adult, it takes about 2 days. And this guy is really easy to take care of. It's really easy to culture. It's very cheap. So this is not a life size. The worm is actually about 1 millimeter long, so it's really small. It doesn't take up very much space. And when the worms are only 5 days old, they already start experiencing neurodegeneration, which is something that we've been kind of toying with in our lab as well. And one thing that Sidney Brenner really wanted to have with his genetic system was he wanted something that still went from a single cell, so fertilized egg, to a have in one that's a hermaphrodite, so it's able to self-fertilize. So this is another reason why I'm using C. elegans for genetics is highly useful. And as I mentioned, it has a very simple nervous system. It only has 302 neurons. And a paper that was actually just released last week, we now have the complete connectome of the worm as well. It's really fascinating. And one thing that we really use C. elegans to its max potential is, in fact, with some in vivo modeling techniques that I'll show. Hopefully, my movies work with this. But we take advantage of the cuticle of the worm is actually transparent. So you can just tag any type of protein with this guy, put it under confocal microscopy, and be able to look at it in real time. And again, we're doing all of this while the worm is in vivo, which is another advantage. And so one thing that I like to bring up as well is in vivo, which is another thing. And so one thing that I like to bring up as well when you're looking at it in terms of a genetic model is when you're looking at genetic processes. This is kind of basic schematic of looking at a mouse cross. And so from one progeny to the next in mice, it takes about months. In C. elegans, we can have a genetic cross in about 2 weeks. That is anything upwards of double, triple, quadruple mutations in this short time as well. So really quick turnover. Okay, so I just wanted to kind of give a basic outline of just some of the major things that I'm going to talk about. If anybody wants to discuss other further techniques later, I'm more than welcome to answer any questions. So again, one thing the Hernley Lab really utilizes is that we can do all of this in vivo in an intact nervous system in real time. And what we can also do is we can also look at single neuronal imaging, which is what we primarily do. But also, C. elegans is really an attractive model for the use of microfluidics, because again, it's very small. And not only does microfluidics help us with different imaging processes, but it also increases our throughput. While increasing throughput, it also increases reproducibility, which is something that's really a big stride right now in the field. But not only can we look at the molecular level, we can also look at the behavioral level. So a lot of what was done kind of in the early stages of C. elegans research was in fact a lot of behavioral assays. And that was really what Sidney Brenner kind of pioneered when he was publishing his first mutations, was actually doing these type of behavioral assays. But now we can take this behavior and we can look at it at the molecular level, and we can also do it in the reverse. Take it at the molecular level and look at how does that produce. Favorite quotes in an interview of Sidney Brenner was he was saying that when he was looking for a system, he really wanted to know that if you had the wiring diagrams, so basically like a neuronal blueprint, could we compute behavior from this? So if I change something in this blueprint, does it compute to change it in behavior? If I do this every single time in the same spot, does the behavior change exactly the same? I just thought that was really fascinating. And again, C. elegans is really nice for this because there's only 300 neurons. So. And lastly, I'll just kind of finish up on a few kind of recent reports and use of genetic screenings and what is probably the arguably the most powerful thing to use C. elegans is for these forward genetic screens. Okay, so we'll kind of start off by looking at in vivo imaging. So this is our confocal here. And what we have here is a nematode worm with a single neuronal process that we look at. It spans all the way from the head all the way to the tail of the worm. This is really important for what we're looking for is because we're looking at glutamatergic transport. And so majority of glutamate receptors are made in the cell body, but then they have to be transported down, upwards of a millimeter away. They have to do this in a relatively rapid response. And again, while the worm is on the confocal, it's immobilized, but it's still alive. So you can take the worm off, put it back on a normal plate, and it lives just fine. There's actually a couple assays where you have to do this, and then you have to put the worm back on for multiple rounds of imaging. So the worm is alive. This is an intact nervous system. So how do we look at, say, glutamatergic transport? Well, what we have here is we have GLUR1 that is tagged with M-sharing. We basically go into a specific to this neuron. We photo bleach a certain region, and then we take a frame-by-frame picture, which then constructs onto this movie. And it looks like everything is playing. Cool. So what we have here is we have, as I mentioned, GLUR1 transport. So we have nice anterograde and nice retrograde transport here. This is just kind of in a wild-type setting. And I know what you might be thinking, okay, this is a small little worm. How are glutamate receptors related in terms of vertebrate research and human research? Well, you can actually take many human invertebrate proteins, knock out whatever that is in C. elegans, put back in the vertebrate homolog, and it will actually rescue activity. So this is really what Fred did in his 2013-2015 papers, where he knocked out CAMK2 in C. elegans, put back in vertebrate CAMK2, and completely rescued glutamatergic transport. And so what we can also get to and rescue transport. And so what we can also do is we can look at cell activity in vivo. So this is just kind of a pretty basic GCamp video in neural culture. Again, just kind of a pretty basic picture on kind of what's going on in culture. But then we can utilize this for C. elegans as well, again, while it's still alive. And so this particular neuron that we study here, one of its major components, besides the fact that it's extremely responsible for memory of the worm, but also it's responsible in controlling backwards movement of the worm. So these worms are actually trapped inside a microfluidic device, but you'll see that when they try to reverse, you'll see this neuron light up, fluorescence, but then once it kind of is stopping, then you don't have any fluorescence. You see here the worm is trying to reverse, now it's not, it's just kind of wondering what's going on. And then now it's reversing again, trying to reverse, and so now we have this process being illuminated. So you can imagine the type of questions that we can utilize this for in terms of how does calcium activity change depending on whatever type of mutation that we are looking at. And so something that's a little bit more sexier that we can use as well is use of optogenetics. So I just decided to look at the TULIP system here, but you can use optogenetics for a vast range of different types of assays in C. elegans, but the TULIPs one is one that's relatively new in C. elegans. So TULIP stands for tunable light-inducible protein sequence. And essentially what you can do is you can artificially recruit proteins together. But in the case of, well, it's kind of how we want to utilize it in the lab, is you can utilize it to artificially induce transport of proteins, of receptors, of organelles. So how this works is you have some type of, you have a very specific peptide sequence here that in the absence of blue light, it's essentially folded in on itself. It's kind of like a clamshell. But then once you shine blue light onto it, it'll actually pop off, where then it'll bind to some engineered PDC domain, which has very high affinity to it. And so not only do you kind of get recruitment of this, but how they're utilizing it in this movie here is they're looking at mitochondria transport. So what you'll see in the movie is we have mitochondria that has the peptide sequence here. Then in the absence of blue light, they're basically not moving. But then once you shine blue light on to the system, then kinesin is then recruited and then they're transported out. So it's a little bit small, so hopefully you guys are able to see it. I have to play it a couple times. See, there's nothing going on, but then they shine blue light, and now not only do you see the kinesin that is tagged, but you also now see the mitochondria. So not only now do we have a system where we can control recruitment of proteins to themselves, but now we can also control things such as transport. And so there are a multitude of questions that you can now ask with this system, and really it's only kind of hindered by your creativity. And so another really powerful tool, so I talked about how, okay, well now we have recruitment of these proteins together, we now have transport, but what if we want to specifically destroy some type of protein, not only locally, but only that protein, and not knock it out entirely, or PALI for short. And so basically what this does is you have some type of protein that you wish to degrade, get rid of. In this case here, which was really illuminated in Fred's 2015 paper, is we have MK2 with a type of mini SAW tag, so mini singlet oxygen generator. So essentially what happens is when you shine blue light onto wherever this protein is, what happens is the mini SAW tag actually creates different types of reactive oxygen species which locally destroy the protein that's closely tagged to. And only that protein as well, which is the key for that, doesn't just kill everything. And so kind of what Fred did was they wanted to find out how if you just locally destroy KMK2 in the cell body, how does this affect glutamate receptor transport out of the cell body? And so here, in the just kind of typical chymograph, you have really nice transport mode in the anterior gradient. You have really nice transport mode in the anterior gradient and retrograde sections, which are eliminated by these kind of traces. But now it is shining blue light onto the cell body, which is now destroying all of the MP2. And you go and you image that same exact area again. What you see is that transport is just significantly decreased here. But not only can you do this in kind of in a large area, but you can also do this very locally as well, very acutely. So what they also did in this paper was they went to a very specific single synapse of the worm, of this neuron, had blue light shine directly onto it, but only in that small little area. And they were able to see how exactly does now removal of receptors from synapses change when CAMK2 is absent from these synapses. And so it is very powerful stuff, but one of the problems with C. elegans and kind of using these really cool tools is it takes a lot of time. So it takes me about 10 minutes to image one worm, and this is either doing transport or looking at just synapses. So you can imagine that when I want upwards of 40, 50, 60 images, it would take a very long time. So something that we've really been trying to engineer in participation and collaboration with Chuck Henry of chemistry department is actually developing microfluidic devices to increase our throughput here. So Hung Lu out of Georgia Tech is really the person that has pioneered and engineered just incredible devices that we have are trying to utilize, but also adapt to our own needs that we need for them. So as I mentioned, it takes me about 10 minutes per worm. Well, what if I told you that I could just flow in 150 worms into this device, which is no bigger than a cover slip? I tell the computer and I program the microscope to say, okay, I want you to go here, image here, image here, image, just move along. All I have to do is press a button, and then I come back in an hour and I have 150 videos that I can sort through whether or not the computer or not. So this is something that we're really trying to do to increase our throughput. And as I showed you that video, the GCAM stuff, which is kind of Rachel's project right now, the other graduate student in our lab, she's able to actually get this down pretty well so far. So things have gone well. But also what Hung Lu and colleagues have really done is they've taken microfluidics now even to the next level. And so I believe she called one of these devices, I don't know if it's one of these, but I believe she called one of these devices, I don't know if it's one of these, but they called it the Worm Hotel. And that is because you can actually sort worms depending on whatever parameter you're looking into. So let's say you wanted to look at some type of screen, or let's say you just wanted to look at something like neuroregeneration in C. elegans. Well, let's say I flow in just a bunch of worms here. They come into this channel, trapped now. You have a laser that can sever the neuron, your neuron of interest. And now it goes into some other different type of sorting device that says whether or not this actual process was successful. So let's say it's looking at this picture and saying, okay, well, this surgery did not work. I'm going to shunt it over on the left side. Now this one did work, and so now I'm going to shunt it to the right side. So then you can just take all those worms we had successful surgery, and you can either leave them there in the hotel, or you can take them out and have any type of mutations that help with neuroregeneration. And another kind of, I guess, simpler sorting device that you have as well that you can imagine using is, let's say you're trying to do some type of screen that either decreases the amount of fluorescence of a certain protein that you're looking into, or maybe potentially increasing this fluorescence. Well, what you can do is you can just take a bunch of worms, blow them through, and now you'll have this fluorescence sorter where basically it'll say, okay, presence of fluorescence, absence of fluorescence. Again, you kind of take these worms and then potentially send them off for sequencing or culture them and keep them going. This is really kind of at the molecular level, but what about behavior-wise? So when we use behavior with C. elegans, we absolutely can So hopefully these movies will work. Okay, maybe not. But that's okay. These ones aren't terribly interesting. One actually had a really annoying voiceover that I ended up playing. So actually kind of worked out. But I'll kind of touch upon kind of what these assays were showing. So we can look at mechanosensation. So a lot of, again, as I mentioned, what Sidney Brenner and the early people that were looking at C. elegans to find mutants and use these forward genetic screens used a lot of touch assays. So what you do is you just take a glass pipette, you actually take an eyelash from your eye, and then you just tape it onto your glass pipette. The reason I use an eyelash is because it's just the right amount to get kind of the harsh touch. Anything a little bit harder, the worm just doesn't really respond that well to it, or can actually just hurt it in general. We use an eyelash, which is kind of funny because if you go into our lab, we just have a bunch of tube racks with glass pipettes and just a bunch of eyelashes on them, so it's kind of funny. But these studies are actually really difficult. You can imagine that if you're looking underneath the microscope, not only do you have to kind of follow the worm because they can be fast, but now you actually have to take your pipette with your eyelash and kind of gently stroke it. And so what is happening in this movie is you have this worm kind of stroke it kind of right below the head, and the worm reverses. And so the parameter of the worm actually reversing you can use for different things. So the reversal behavior has actually been linked to glutamatergic transmission in that long neuron that we're studying. So let's say you go ahead and you give the worm a nice stroke and it just doesn't do anything at all. It just keeps on going. Then maybe what is happening is we have impaired a glutamatergic neurotransmission. This was actually how one of the glutamate receptor mutations was actually found in C. elegans in the lab where Fred was doing his postdoc. But then there's also other ones. So let's say you're looking at sensory neurons. So what we have in these two videos is essentially you have some type of repellent of the worm. And what you do is you drop it on the tail of the worm, and then just kind of by general pressure, it'll actually just go up to the head of the worm and just kind of encompass it. And what will happen is if it's something that the worm doesn't like, again, repellent, then the worm will reverse. But if it's some type of buffer, like just a salt solution that they like, then it doesn't phase them out. It's going to keep on wiggling forward. And again, we can now use this to say, okay, well, maybe it's some type of sensory transmission that's impaired onto this glutamatergic neuron that we study. That's impaired onto this glutamatergic neuron that we study for this. But can we now take this behavior to do a lot of more practical things, a broad general scheme? And you can using this chemotaxis assay, which we'll kind of talk about now. And we use this chemotaxis assay to actually study memory in worms. So not a lot of people know it, but worms do have memory. They're capable of learning. They have short-term, they have long-term memory. And so what we do is we subject them to this experiment where we just take a bunch of worms, we put them in the middle of the plate, and then on either extreme, we either have some type of chemoattractant. So in this case, we have diacetyl, which is something they like, they love, they'll just go right towards it, or some type of control. So in this case, it's ethanol. And so we just take these naive worms from a normal plate onto this, and the majority of them will trust. You don't really have to know what this chemotaxis index really stands for. You just have to know that the higher this number here, the more attracted they are to this diacetyl. So what we want to do is we want to now study if their learning behavior. So what we do is we take a bunch of worms, we put them on a plate with no food, and we put a drop of diacetyl on top of the plate so they can smell it, but they can't actually reach it. So you let them go for an hour, and then after this hour-long period, they start to associate the feeling of starvation now with the smell of diacetyl. So if you now put them on this same experiment and you say, okay, well, do you like the diacetyl or do you like the ethanol? Majority of them say, okay, I don't like the diacetyl. I'd rather go to the ethanol because I don't want to get starved if I go over here. And now to really look at, so this is more on the learning paradigm, but what about memory? Can they retain this? And what you do is you take a different batch of worms, you condition them for this 1 hour starvation period, then you put them back on a plate with normal food so they're nice and happy. You let them go 4 hours for short-term memory, and then again you let them go on this assay. And what you'll see is that majority of the worms, they do remember just fine. They say, okay, although it's been this amount of time, I still remember that I don't want to go towards this diacetyl. I want to go more towards this ethanol. But some worms don't really get it. They're like, no, I still like that diacetyl. I'm going to go towards that. So we can use this to study memory. This is actually one of the proteins that I've actually been studying. It's phosphatase that when you knock it out, it seems to have impaired short-term as well as long-term memory, but it learns just fine. It's capable of learning, it's just able to capture and retain its memory. What you can also do is you can do this on ale. So what you do is you condition the worms twice with 30-minute intervals where they're allowed to be on food, nice and happy. And you let them go 24 hours, and again, you do these same experiments. What you find is that, again, some worms remember just fine, even after this 24-hour period. They say, no, I still remember that, I don't like that diacetyl. I'm going to go towards that ethanol. But again, we have some worms which are almost essentially like the naive, where they are still extremely attracted to diacetyl. They don't remember at all. So is it possible now that we can take this scheme? Can we go from the behavioral level to the molecular level? Well, we can. And this is something that we've been working on with a couple of our collaborators in Switzerland, where we basically take worms that have been subjected to this experiment and we them from either diacetyl or ethanol. We look at their surface levels of glutamate receptors, because again, what we're really interested in is glutamate receptor transport, as well as synaptic glutamate receptors, and really how that is related to memory. And so again, what we can do is we can take the worms that have conditioned and presumably learned this, take them from either the 2 drops, we can look at their synapses and we can look at their transport and we say, how does this differ now between naive and learned? And so surprisingly, well, I guess not surprisingly, but interestingly, what you find is that, so in the naive, we're looking at surface synaptic WER1, so that's what we're looking at here. This is WER1 with an SCP. So just looking at surface level WER1. In the naive worm, but then when you look at the conditioned worm, we actually do see this, which is something that is almost like a no-brainer, like duh, of course this happens, but it hasn't really been shown on this level kind of what we're looking at here. And to say definitively, although we aren't saying definitively yet, we still have a bunch more that we need to do with this. And so as I mentioned, kind of arguably one of the very powerful things that you can do with C. elegans is using them in forward genetic screens. Basically what you do is you just take a large amount of worms, expose them to some mutagen, EMS, you let them go a few generations. And again, this would only take about 5 days, not even 4 days. And then you're either looking for either the absence of a phenotype or potentially the presence of some other phenotype. And then again, you can kind of keep these guys going, you can send them off for sequencing and kind of see what's different. But then I kind of briefly touched upon the fact that RNA is strong in C. elegans, and all you have to do is feed them double-stranded RNA, and it works just as good. And so C. elegans, they feed on E. coli. So you can imagine it's pretty easy. All we have to do is engineer E. coli to express our double-stranded RNA. Once we eat them, it gets incorporated into our system. Now we have gene silencing. So if we have some type of RNAi library here, we can put the worms on them, do a bunch of phenotypic screens, and then potentially confirm and validate the CAP. So one thing that I found when I was looking in a relatively kind of recent paper is actually using C. elegans in ALS studies. So apparently there are a lot of labs that use C. elegans in ALS studies, and that is because the same quadruple G double C repeat in humans, if you engineer that into C. elegans, they're pairwise. So the same quadruple G double C repeat in humans, if you engineer that into C. elegans, they're pairwise, so they don't move. You can imagine then you can do a type of screen where you have these mutants, subject them to some mutagen, and in our progeny, if we find worms that are moving just fine, then maybe potentially we could have found the suppressor of this repeat. That's exactly what they kind of find and they touch upon in this paper. And so what if you want to look at something else? What if you want to look at, well, let's say motor neurons? Where you can do this other type of assay where essentially they're doing the whole mutagenesis, they're letting them go. And then what you have here is this outer ring assay where you have some type of chemoattractant, like diacetyl, on the outer edge, and you put the worms in the center, and the worms that are unable to make it there because of deficiency can't move. Again, you can take these guys, culture them up, look at their mutations, and see if there's anything different. And one that I thought would be interesting for this group is this paper that came out in 2014. Again, this is with our collaborators from Switzerland. And many times when we're kind of looking at memory, we like to think of kind of strengthening memory, keeping memory. How do we make it better? Well, actually what they found was this protein called MSI1, which is part of the Musashi family. And they find that it's actually responsible in forgetting memory. That if you have this knocked out, what you have is what they call super memory. The worms remember for extended periods of time. So that chemotaxis assay that I told you about, where the worms are kind of deciding whether or not to go to the diacetyl or the control, these worms with this knockout here have extreme super memory, which is really interesting. And I highly recommend that you look this paper up. And at the end, I have all my references and everything. I believe these are online, so feel free to look that up and read that. It is really interesting. So I just kind of wanted to finish off on what the Hermle Lab is doing, just to kind of give you guys an idea of some C. elegans neurobiology research that is going on here at CSU. So many of you might have talked to Fred or been to one of his talks or anything. Seen him in the hallway, he'll talk your ear off. But the basis of kind of how the Hermle Lab was founded was Fred was studying this molecule, calcium calmodulin kinase 2, or CAMK2. He was really looking at how its role in synaptic plasticity, because before this, many people in memory, it was studied solely at the synaptic level. Which makes sense, right? That's where the receptors are, that's where all the magic happens. But Fred's kind of argument with this was, well, although a lot of interesting things are happening here, we have all these upstream processes that have to happen for the receptors to even just get there. And so how is it then that a receptor that's primarily made in the cell body knows that it needs to go to synapse 2 and not synapse 3000? Or how does it know it needs to skip all of those synapses, go to synapse 1300 and not synapse 2? This was really kind of the foundation of the Hermle Lab here, and we're really interested in looking at how different parameters change glutamatergic transport, and really how that relates to synaptic plasticity and memory. So a really cool project that's been happening is one by Rachel in our lab. She's looking at reactive oxygen species. And the reason that she started on this was because Fred touched on it a little bit in his 2015 paper, where they were looking at a type of catalase, so presumably increased reactive oxygen species, and how that is kind of working in this pathway of glutamate receptor transport. And what Rachel's actually been able to find is that when you do have increased ROS levels, you have decreased transport here, which thinks it has something to do with the calmodulin pathway with this. I won't spoil it too much. Hopefully you guys can kind of keep up with that project, because we're hoping to get that one and mine out here pretty soon. So then this is one I've primarily been working on. So again, it's mentioned, Fred has primarily worked with CAMK2. So when he started the lab, he asked me, he said, hey, do you want the, this is when I was a rotation student, but do you want the easy or do you want the hard project? And this is when I was a rotation student, but do you want the easy or do you want the hard project? And of course, I said the hard project, but hindsight's 2020. But here we are now. But what he wanted me to look at was looking at phosphatases in this regulatory pathway. If you look at CAMK2, it's a kinase. He thought, well, it's more than likely that some type of phosphatase is working in this pathway as well. So he gave me a list of all of the known phosphatases in C. elegans, and he goes, here you go, pick one. So I did a bunch of literature background, and I found this one. It's a tyrosine phosphatase. And what I've been able to find is that in the absence of this phosphatase, it kind of has some type of dual role in this pathway where it is somehow controlling transport of glutamate receptors such that if you don't have this phosphatase, you actually see significant decreased transport, but then at the synaptic level, when you don't have this phosphatase as well, it seems that the receptors are just unstable. They get there, but then they're just immediately removed. They get there and they're immediately removed, which is what we kind of think is happening in the memory paradigm that I mentioned, that you don't have this phosphatase, you have decreased memory, both short-term as well as long-term. We think it has something to do with this. But again, if you guys have any questions about anything I've talked to you or just worm stuff in general, I'm always happy to talk about it. And thank you, Jim, for allowing me to talk today in the absence of Fred. And again, here is all my references and just kind of some really cool website that you can do with the worm. And what's also really nice about this is that we just have an extremely large database of everything with the worm. If you want a specific mutant, all you have to do is go onto a website that is stationed out of Minnesota, pay $7, and there you go. You now have a mutant. It's really good on Wormbase, and it's just an awesome organism to work with, in my opinion. But thank you. Yeah. Right. In terms of addiction studies, C. elegans has been used, not terribly, but that is something that I actually brought up to Fred when I was looking at a proposal for my prelim. It was actually using C. elegans in like addiction type research and saying, how does this kind of relate to this glutamatergic pathway in terms of that research and saying, how does this kind of relate to this glutamatergic pathway in terms of that. Yeah, that's a great idea.
So I actually have a question, but do you guys also look at the LTP at the synapse level? Like, do you see at a particular synapse that is getting more adversarial insertion?
Yeah.
The whole
Yes. So in the lab here at CSU, we have not done any type of electrophysiologic.
You don't just I'm not just SCD2A1, for example, it's surface localization and aggregation at a particular synapse versus overall.
Yes. So this is also something that we've been really trying to get at, and what has been a lot of critique with some of our grants is saying, well, if you look at this process that we have, so kind of like our general picture, how do you know that this illumination spot? What we've been trying to do is we've actually been trying to do either like split GFP or GRASP system, where we have particular neurons that we know are presynaptic to our specific ABA connection, and potentially what we can do is now we can use this split GFP system to now look at and say, okay, well, if this spot is illuminating, only this spot, so we know this is a synapse.
But why can't you just simply do like staining for postsynaptic markers? Is that like you kind of just...
That's the problem is, we don't have PST95, so that's one of the main downsides of using C. elegans. Is some of the
what about NMDA receptors? You also get like an NMDA dependent
Yes, so in terms of the NMDA response, it is similar to kind of what you see in vertebrate systems, but its pathway is a little bit different in terms of its transmission rate.
There's no immediate.
Well, no, there is. But what's weird is that it was long thought that action potential propagation is only sodium dependent. And that it was long thought that in C. elegans that they didn't have action potentials, but actually recent papers have come out where they've shown that action potentials